Okay, I was going to go back over the Amoeba Sisters video recap with you. The first box says, in the box below, please illustrate an enzyme and substrate and label the following keywords in your illustration. Well, for the enzyme, we can just put something that looks kind of like the little Pac-Man that they were talking about. Excuse my drawing on the computer, but we'll get there. So there we go. And this will be our enzyme. That's our enzyme. Enzyme. And then we can maybe use a different color. Nope. Okay. So this one right here that fits right into our enzyme. It's like a lock and key mechanism made just to fit perfect. The enzyme fits perfect with the substrate that it's going to work on. So this right here, this would be our substrate. And that'll be our substrate. Okay, the substrate is the reactants that are going into the equation. I'm just going to leave it as sub there. It'll be our reactants that are going into the equation, the ones that are on the left side of the arrow generally, and the products, the arrow points to the product. They both start with P, so the arrow points to the product. So our enzyme, and then our substrate, and then right here, this area right in here where they stick together. Okay, let's see, I can maybe that right there. That area where they're stuck together, that is what's known as the active site. So that's where all the activity is taking place. It's the active site. So the active site is where the enzyme and substrate are bound to each other. So that's the active site. So you can draw something kind of like that. I'm opened in DocHub. So if you open in DocHub, you should be able to do what I'm doing here. It's not real pretty, but it gets the point across, I hope. So the enzyme, remember the enzyme does not change. It's going to bind to this substrate. And then the reaction is going to take place. If it may be a big compound that the enzyme is going to break apart. And then it would release it as two separate things into a new product. So you get the enzyme substrate complex. That's what it's called when they're stuck together. It's the enzyme substrate complex. The substrate will change and come off as a new product. The enzyme will not be changed. The enzyme will stay the same. It's not used up. It's still there. The substrate will come off of it after the reaction is finished. The enzyme will be unchanged. And then it's able to bind another substrate and carry out the reaction again. So the enzyme can be used over and over again. It does not change. The substrate will come off as a new product. Okay, number 2 says enzymes are typically which type of biomolecule? Well, we've talked about carbohydrates. We've talked about proteins. We've talked about nucleic acids, and we've talked about lipids. So which one does the enzyme belong in? Okay, most enzymes are proteins. Okay. Most enzymes are proteins, so we can put proteins right here. Oops. Most enzymes are proteins. And then it says, describe the effect that an enzyme has on a substrate. Well, the enzyme, like we just drew, the enzyme binds the substrate, lowering the activation energy. Lowering the activation energy is important. That's the main mechanism of the enzyme. This is not exactly how it happens, but this is how I think about it. I think about you have two different chemicals running around, maybe inside a cell or in a petri dish or somewhere. They're all running around, and they're having to chase each other down to get together to have that reaction. Well, the enzyme is going to catch those, just like it fit over here in this thing. The enzyme's going to catch those chemicals, and it's going to hold them in place so they don't have to chase each other anymore. They don't have to use as much energy. So the enzyme is lowering the activation energy. So the enzyme binds the substrate, lowering the activation energy. Then it will allow the reaction to occur. At a faster rate. And sometimes that means it's either putting together two chemicals as part of the two parts of the substrate to make a new product, or sometimes there may be a substrate there and it's going to break it apart and make a new product. Anyway, the enzyme is going to bind to the substrate, lowering the activation energy. I should say energy there. Oops. It's going to lower that activation energy. And then it will allow the reaction to occur at a faster rate. Learning curve here, I'm getting there. Okay, so allow it to occur at a faster rate. And that's that one. Then we have down here, it says, in order to function efficiently, enzymes need to be at an ideal pH and temperature. Different enzymes have different ideal pH and temperature conditions. Like we talked about yesterday, that most enzymes in our body are going to work around 7.1, around that neutral, because we're mostly water, and then our blood also has a pH of about 7.1, and so most of our body is kind of close to that neutral. Until we get to the stomach. We said the stomach is really acidic because it's got that hydrochloric acid in it that has a pH of about 2, so it's very acidic. Remember, acid below 7, A's and B's go together, so acidic below 7. So it has a very low pH. That will be one place that you would see enzymes that work at an extremely low pH instead of that nearly neutral pH. So it's that optimum pH and temperature are going to be different for different enzymes in different places. So but it says if the pH or the temperature is extreme for a particular enzyme, it can even denature the enzyme. And that's like when Pac-Man gets bitten by the ghost. It goes boop, 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 and Pac-Man opens up. Well, it's kind of what happens when it's denatured. When an enzyme denatures, its shape changes. Something happens with the bonds and the shape changes in that enzyme, and then it's no longer, it's like a broken lock. The key from the substrate no longer fits in there. So denatured means that something has caused its shape to change. Its conditions, environmental conditions have caused the shape to change. It has denatured. So it denatures an enzyme which can prevent it from binding and acting on its substrate. For the following two scenarios, name the variable temperature or pH that is affecting the function of the enzyme. So our answer is either going to be temperature or pH, right here. Temperature or pH. There shouldn't be anything else. It's going to be temperature or pH. And pay attention to these questions. There's a possibility you can see them again. And we do have a test coming up on Friday. So temperature or pH. It says ATP is produced by cellular respiration in human body cells. There are a variety of enzymes that work to produce ATP, but one of those enzymes is called phosphofructokinase 1. This enzyme is sensitive to blood acidity. Blood can become more acidic if a patient is in respiratory distress. So it said acidity right here, and it said more acidic right there. So if we're talking about acids and bases, we're talking about pH levels. So that would be pH that's changed here. So that would be a pH that is inhibiting that enzyme from working. So pH would be the answer there. B says a popular lab that can be performed by students is to test the reaction rates of catalase enzymes when it acts on the substrate hydrogen peroxide. Catalase has the ability to break down hydrogen peroxide. Catalase can be found in beef liver from the grocery store. However, if beef liver is boiled, the catalase will not be able to break down the hydrogen peroxide. Okay, so in this scenario, what variable is affecting the enzyme function? Would it be pH or would it be temperature? Well, it didn't tell us anything about anything being acidic or basic, but it did say that you're going to boil that beef liver. So boiling would be at very high temperature. So in this case, temperature would be your factor. Temperature on that one. Okay. And down here, we're just going to look at the enzyme and substrate real quick. We're not going to worry about the drawings and things. So lactase is an enzyme that breaks down sugar found in dairy products, known as lactose. Some people are lactose intolerant, and this can be due to not having enough lactase production. People who are lactose intolerant may not feel well after eating food containing lactose. Now, we said generally enzymes are going to end at ASE. So right there it is, lactase, L-A-C-T-A-S-E. Lactase would be our enzyme, and it tells us this, acting on the sugar lactose. Ose means sugar. So for our enzyme, we would put lactase. And they had already put lactose in there for us. So that's that one. And the illustration would just be much like the one we drew up there. Next, this one says an enzyme called glucocerebrosidase breaks down a glycolipid in the body known as glucocerebroside. However, in the genetic disease known as Gaucher's disease, the body does not produce enough glucocerebrosidase. Therefore, the glucocerebroside can build up in the body, and this can cause serious side effects such as anemia and swelling of the liver and spleen. So again, we're looking for the enzyme, and it even told us an enzyme called glucocerebrosidase. Let me what that is. So I'm going to slide my box over because that's a really big word. Glucocerebrosidase. Let's see if I can make that a little bit smaller. Maybe it'll fit in there. There we go. And then our substrate, this time it's not a sugar. It tells us this time it is a glycolipid, so it's not going to end in ose, but it would be glucocerebroside. And that's going to go right here. Glucocerebroside. And again, our drawing would look kind of like that little Pac-Man image. So pancreatitis is an inflammation of the pancreas which can damage pancreatic tissue. The pancreas produces digestive enzymes such as amylase and lactase. These enzymes assist in the breaking down of certain food biomolecules. In this disorder, enzyme productions from the pancreatic tissue may be stopped. Okay, so again, our enzymes, we have two this time. We have amylase and lactase. A-M-Y. L-A-S-E, and lactase, L-I-P-A-S-E. Okay. And thing about the names of many of these enzymes is they tell you what they're working on, like glucocerebrosidase works on glucocerebroside. Not spelled correctly there, but okay. That one should be glucocerebroside. And then lactase works on lactose. And here we have lipase. What do you think lipase might work on? Because it didn't tell us. Lipase actually breaks down lipids. Okay, amylase is a little bit different. It doesn't really have the name of what it's breaking down, but we talked a little bit about that yesterday. We said it's in our saliva and it begins breaking down starches in the mouth. So amylase is going to break down starch. That's that one. Okay, hopefully that'll help. I will post the study guide later. We're doing it tomorrow, and I'll post up the study guide answers later tomorrow evening. So you'll have to make sure you have the correct answers to study. We do have a test on Friday, so please study that. Thank you. Y'all have a good evening.